Hi all and welcome to the Gemophile. Have you ever come home with mud on your shoes and your parents tell you to wipe it off? That ordinary looking dirt might be hiding the ingredients for life-saving medicines. In this video, we're going to zoom into a handful of soil and see how scientists are using it to hunt for new antibiotics to fight dangerous superbugs. Antibiotics are drugs that fight bacterial infections by either killing bacteria or stopping them from growing. They've helped turn infections that were once deadly, like pneumonia and infected cuts, into problems doctors can often treat in just a few days. Bacteria, however, are constantly adapting. As time goes by, they can develop mechanisms that enable them to withstand these medications. For example, they can pump the antibiotic out of their cells or cut it up with enzymes or change the target the drug was supposed to hit. When that happens, we call those bacteria antibiotic-resistant or superbugs. Health organizations like the World Health Organization and the Center for Disease Control, commonly referred to as the CDC, warn that millions of infections every year are now caused by bacteria that are hard to treat, and tens of thousands of people die from them. This means that new antibiotics are needed. Here's where the soil comes in. Many classic antibiotics come from soil bacteria. A famous example is Streptomyces. This is a group of bacteria that live in dirt and give off that earthy smell after the rain. Scientists grew these microbes in the lab, tested the chemicals they made, and discovered drugs like streptomycin, which is used in the treatment of diseases such as tuberculosis. For a long time, that was the basic recipe. Collect soil, grow the bacteria on plates, see which ones kill dangerous microbes, and purify the useful compounds. But there's a huge problem. Most bacteria in soil simply don't like growing in our lab conditions. Imagine trying to keep a deep-sea fish alive in a bathtub. If we can't grow those bacteria, we can't easily test what they make. That means a lot of potential antibiotics stay completely hidden. What if we don't try to grow the bacteria at all? What if we just read their DNA directly from the soil? This is where modern DNA sequencing in a field called metagenomics come in. Step 1. Scientists collect soil from places like forests, fields, or even your backyard. Step 2. Instead of trying to grow each microbe, they break open all the cells in the sample and extract the mixed DNA. It's like dumping all the instruction books from every organism in that soil into one giant pile. Step 3. Powerful DNA sequences read millions of tiny fragments of that DNA. Step 4. Computer programs act like puzzle solvers. They take those fragments and piece them together into reconstructed genomes. Best guesses of what each hidden bacterium's full DNA book looks like. Step 5. Now comes the treasure hunt. Inside those genomes, researchers look for gene clusters that have the right patterns to make antibiotic molecules. Once they spot a promising gene cluster, they can copy those genes into a microbe that does grow well in the lab, let it produce the compound, and then test whether it kills dangerous bacteria, including those that are drug resistant. Using this approach, scientists have discovered hundreds of previously unseen soil bacteria on the computer, without ever growing them on a plate. From those invisible microbes, they've identified new potential antibiotics like erutacidin and trigentamycin, both reported in 2025. These antibiotics have the potential to attack bacteria in fresh ways. First, antibiotic resistance is one of the biggest health challenges of this generation. The more we overuse and misuse antibiotics, the more we push bacteria to evolve resistance. That makes surgeries, cancer treatments, and even simple infections riskier. Second, this DNA from dirt strategy shows how microbiology and computer science team up. It's not just petri dishes anymore, it's also sequencing machines and algorithms. And third, it's a reminder that everyday environments like the soil you walk on are full of unseen microbial worlds that could hold solutions to global problems. So next time you're asked to wipe the dirt on your shoes, consider that the soil under your feet is full of bacteria we may never be able to culture, but we can still read their DNA and borrow their chemical tricks. Dirt becomes a library of possible life-saving antibiotics. To learn more about how scientists discover potential antibiotics from the DNA of soil bacteria, check out the references linked below. That's it for today. Join us next time on the Germophile as we explore the fascinating world of microbes together.